The effects of metabolic toxins on cellular respiration. Oxidative phosphorylation and chemiosmosis produce a total of 32 ATPs in cellular respiration. For these 32 ATPs to be made, the following operations need to take place. NADH and FADH2 carry 2 electrons with them. These electron carriers drop off the 2 electrons that enter the electron transport chain. NADH drops off its 2 electrons at NADH dehydrogenase, while FADH2 drops them off at ubiquinone, skipping NADH dehydrogenase. The electrons enter a transport chain in which they move from lower affinity to higher affinity in order of NADH dehydrogenase, ubiquinone, cytochrome BC1, cytochrome C, cytochrome oxidase, until they are finally taken by the final electron acceptor, oxygen. Oxygen then takes the 2 electrons as well as a proton to produce water. The movement of electrons across the electron transport chain releases energy that is used to pump protons out of the matrix and into the intermembrane space. This creates a high concentration of hydrogen in the intermembrane space and a lower concentration in the matrix, creating an electrochemical gradient. In the process, protons are moving against their concentration gradient and creating an excessive concentration of hydrogen in the intermembrane space. There's a whole lot of potential in the chemical gradient in the proton, which generates PMF, or proton motive force. ATP synthase then makes use of this PMF when it allows protons to flow along their concentration gradient back into the matrix. As it allows the protons to flow back, it uses PMF to bind an inorganic phosphate with an adenosine diphosphate, creating adenosine triphosphate. ATP will then be used for energy used in various cellular functions and activities. The production of these precious 32 ATPs is what keeps cells working and organisms alive. Surely, if something were to disrupt this intricate process, the consequences would be devastating for the cell and the organisms the cell makes up. But really, what would actually happen if an organism were to be exposed to something that disrupted this crucial segment of cellular respiration? Say, for example, the extremely hazardous substance of Antimycin A. This secondary metabolite produced by Streptomyces bacteria is an active ingredient in finchal, a chemical pesticide, which is a fish poison, used in fisheries management. It's often used to control invasive species and also as a fungicide in agriculture to protect crops. Agromycin 50 is a very popular one. Some studies even suggest that it could have potential in cancer treatment by stopping cancer cells from growing. Since it's pretty powerful, it has to be used carefully to avoid unwanted environments and bodily effects. Now let's discuss what makes this substance so deadly. Antimycin A is a potent inhibitor of the cytochrome BC1 complex. Specifically, it binds to the QI site of the cytochrome BC1, where it blocks the transfer of electrons from ubiquinone, obstructing ubiquinone's reduction, preventing electrons from moving along the electron transport chain and being received by oxygen at the end means to prevent the released energy during transportation to higher affinity acceptors to be used to pump protons into the intermembrane space. If protons cannot be pumped out of the matrix, the concentration gradient used to generate PMF will no longer exist. Since the concentration of protons in the matrix and the intermembrane space will not be very different, the protons will have no reason to go through chemiosmosis and flow back into the matrix through ATP synthase because, well, it's no longer along their concentration gradient to do that. Without PMF and protons flowing back into the matrix through ATP synthase will no longer be able to use the electrochemical gradient's potential energy to bond an ADP with an inorganic phosphate and make ATP. This will result in ATP not being produced through oxidative phosphorylation and chemiosmosis. The 32 ATP produced by the ETC are essential to maintain cell livelihood and function, which is why Antimycin A's destructive damage to ETC and thus the mitochondria can result in cell death and in consequence the death of the organism that has those cells. Here are my works cited. Thank you for listening.